Hello. Okay, so here we have a biology paper, February-March 2021, paper 4-2. Okay, so we'll go through the paper and we'll discuss it and see what the questions in this paper actually wanted. Let's start with number 1. Number 1A says, figure 1.1 is a diagram showing the position of some organs in the human body. We can see here we have different organs. We have like organ A, B, C, E, and D. Some of the organs shown in figure 1.1 are endocrine glands. Table 1.1 shows the names of some of the endocrine glands, their identifying letters, and the hormones that they produce. Complete table 1.1. So the table is to be completed by stating the name of the gland, the function of the letter of what? Of the gland. Okay, so we have this which is labeled C. We go back to the diagram. You find that C is found here just above the kidneys. The gland that is found above the kidney is adrenal gland, and the hormone that it produces is usually adrenaline. So we'll state here adrenal as the gland and adrenaline as the hormone that it produces. Then we have insulin. Now insulin is usually produced by the pancreas gland. Alright, now where is insulin? Insulin is literally produced by the gland here, E. Pancreas. So here we write pancreas and the letter is E. The hormone produced is insulin. Then we have the testes. The testes are here represented by letter D. What is their function? Their function is to produce the male hormone known as testosterone hormone. Alright, yeah. So those are the glands together with the hormones that they produce. Okay, B says figure 1.2 shows two graphs representing the relative blood concentrations of two hormones A and B released by ovaries during the menstrual cycle, the thickness of the lining of the uterus. Alright, so we have the first graph over here which is showing the concentration of what? Of hormones over the days, and then we have this one here which is showing thickness of the lining of the uterus over the days. Roman 1 says describe the roles of the menstrual cycle for hormone A. Hormone A is literally this hormone here, and we can see that highest concentration of this hormone is during the first days of what? Of the flow. Okay, so what is this hormone? This hormone here is estrogen. So we'll say hormone A is estrogen hormone. Now, what is the function or the role of estrogen hormone? First of all, we say that it stimulates repair of lining of the uterus. Lining of the uterus, yeah. So this is what the estrogen hormone does. First of all, it repairs the lining of the uterus after menstrual flow, but then again it stimulates secretion of luteinizing hormone. So again it stimulates secretion of luteinizing what? Hormone. Okay, which means it now like promotes production of luteinizing hormone, but at the same time it inhibits secretion of follicle stimulating hormone because it is now stimulating luteinizing hormone so that the egg is released. So if the egg is going to be released, then of course follicles have to stop growing. So here we can say that it inhibits the follicle stimulating hormone. Yeah, so those are the roles of estrogen hormone. Then state the day in figure 1.2 when ovulation is most likely to occur. Ovulation is the release of the egg. So we come here to figure 1.2. Alright, so ovulation is most likely to start occurring here around day 12 to almost day what? 16. Alright, so we'll state here day 12 to 16, any day in between 12, 13, 14, 15, and 16. Most likely it is on day 14. Alright, set the days when the lining of the uterus is lost from the body. So the days of when the lining is lost is definitely during the menstrual days. Alright, so it's expected to start from this point over here until here where we have low percent of thickening of the uterus. Then that is day 0 to 6. So say 0 to 6, whereas after that the lining increases. Okay, C says female contraceptive pills is a chemical method of birth control which is available in many countries. Describe the social implications of increased availability of female contraceptive pills. Alright, so social implications can be both negative and what? And positive. All right. One of the positive ways is that it gives a chance for women to engage in other activities such as education, all right, to get employed on different posts, okay. But also it can have negative effects. Negative effects could be such as increase in spread of STIs, because people could just engage in social activities and of course they don't care because they'll not get pregnant, all right. So increase in STIs, but also it could lower birth rate, all right. Yes, so some implications could be such as lowers birth rate, okay, but also we can speak of increased spread of STIs, all right. Yes, but also increased chances for women in employment and education, all right. Those could be the implications of female contraceptive pills. All right, the hormones in female contraceptive pills can enter rivers. Describe the negative impacts of female contraceptive hormones entering rivers and contaminating drinking water. So what will happen when these hormones enter into drinking water? The first thing, we expect reduction of fertility in both males and females because they are going to use the water as drinking water, all right. These males and females could be the organisms in water or humans who use that water, okay. So if we have decreased or reduced fertility, then we can have reduced number or reduced population, number of organisms that have used or have been affected by these contraceptive pills, all right. Yes, so we can say here that the impacts will be reduction of fertility in aquatic organisms, reduction of fertility in aquatic organisms, but apart from that, we can have reduction of fertility in humans, all right. Yeah, because humans will also use that water. So reduction of fertility in humans. Apart from that, we can have a reduction of population, all right, reduction of population because the fertility rate has decreased. So we can have reduction of population in aquatic organisms. Yeah, so these are the effects of of female contraceptive hormones entering into rivers and contaminating drinking water. Then stage 2 barrier methods of contraception. So barrier methods of contraception, they are meant to prevent sperms from fertilizing the egg or from reaching the eggs. Now one way which can be used is by a female inserting a diaphragm like stuff, which is going to prevent passage of sperms. So we have the diaphragm, but also use of condoms, all right. So condoms also can be used to prevent sperms from reaching the eggs. All right, we move to the next question. It says here 2.1 is a diagram of a prokaryotic cell. State one visible feature in 2.1 that determines this as a prokaryotic cell. Now when we speak of prokaryotic cells, we say that these are cells that do not have a nucleus, all right. So when we look at this here, we see that it has a loop of of DNA, which is not in the nucleus, all right. But also it has plasmids, which you can find them in prokaryotes, all right. Yes, so we can say here the DNA loop is not surrounded by nucleus, not surrounded by nucleus, or you can say it has no nucleus. When you say it has no nucleus, it proves that this is a prokaryotic cell. State one other cell structure that is present in cells of all organisms. Now, what do we actually find in cells of all organisms? We find cytoplasm, we find cell membrane, all right? Yes. So we can find cytoplasm. We can just name cytoplasm as one organelle that we find in all cells. Prokaryotes, animals, and plants are 3 of the 5 kingdoms of organisms. State the names of other 2. Here we have prokaryotes, we have animals, we have plants. Now, what are the other 2 kingdoms that are not stated here? We have kingdom fungi and kingdom protoctis. They are the ones that are not stated over here. So we'll write fungi and we'll write protocysts. Yes, so those are the two kingdoms that are missing. C says, figure 2.2 shows parts of the nitrogen cycle, and we can see it over here. The cycles, like we have nitrogen in the air into nitrates, we have nitrates in the soil into nitrogen in the air. Yes, so that is part of the cycle. Then describe processes A, B, and C. Let's start with process A. When we look here, we can see that process A is actually coming from dead organisms forming what? Nitrate ions. Alright, so this obviously involves decomposition of the dead organisms into ammonium salts, okay, or ammonium ions. So ammonium ions will then be converted into what? Into nitrates under the process of nitrification. But then again, we have process B, which is forming nitrate ions directly from nitrogen in the air. This process is known as nitrogen fixation, and of course it is done by what? By bacteria. And we have C over here, which converts nitrate ions into what? Into nitrogen in the air. This process is known as denitrification. Alright, so we'll say here, process A involves decomposition to produce ammonium. Involves decomposition to release ammonium. Okay, then ammonium undergoes denitrification to form nitrate ions. Yes, that is the first thing. Then process B is nitrogen fixation. Okay, nitrogen fixation. Whereas here we say that atmospheric nitrogen is converted into nitrates. Nitrogen is converted into nitrates by either lightning or nitrogen fixing bacteria. And process C, we'll say this is denitrification. Okay, so this process here involves conversion of nitrates into nitrogen gas. Okay, so it involves conversion of nitrates into nitrogen gas. Yeah, so this is what is taking place in this diagram shown over here. And then Roman 2 says, state the name of the process that plants use to absorb nitrate. Alright, the name of the process that plants use to absorb nitrates is usually called active transport. So we'll say here active transport. Alright, then we go further. Number 3 says the activity of the heart can be monitored using different methods. Figure 3.1 shows 2 ECG traces. One trace was recorded when the person was at rest, and the second trace was recorded when during exercises. The length of the time for one heartbeat is indicated in figure 3.1 on the ECG trace recorded at rest. Alright, so here we have the ECG trace at rest, and this one here is during exercises. Alright, and then we have a heartbeat rate recorded over here. Okay, yes, so we can see that from here to here, this is one heartbeat. Yes, here, this is one heartbeat. Alright, so from here to here, it has taken 0.2 seconds. And then from here to here, it has taken 0.2 seconds. And here to here, it has taken 0.2 seconds. So from here to here, it's approximately 0.6 seconds. And then we have from here to here again. Yes, so this is approximately like 0.6 until here. 6, 5, let's say. 6, 7. So it's approximately 0.68, approximately 0.68. So from here to here, the time taken is approximately 0.68 what? Seconds. All right, yeah. So the next question says, estimate the resting heart rate for the person from the ECG trace. So heart rate will be taken by the amount of time we divide it the number of heartbeats during that particular time. Alright, so let's assume we are going to check the heartbeat rate in 1 second, which is 60 seconds. But in these 60 seconds, we know that the heartbeat is taking what? 0.68 seconds to do what? To beat. Alright, so how are we going to do it? We know that in 1 minute, which is 60 seconds, the heart beat like almost 0.68 times. It took 0.68 seconds for the heart to complete what? 1 heartbeat. So 0.68. All right, in 1 minute or 1 heartbeat, for a length of 1 minute, it took 0.68 what? Seconds to complete 1 heartbeat. So we'll take that time, which is 1 minute, divide it to what? To 0.68 seconds, which is the time of what? Number of heartbeats in this one time. So when we divide these two here, we get yes, we get 88. We get 88 beats per minute. So when we take the time taken, we divide it to the time taken, which is 1 minute. We divide to the number of heartbeats. Okay, in this 1 minute, we get the heartbeat per minute, which is definitely going to be 88 beats per what? Per minute. Okay. Roman 2 says, explain why ECG trace recorded during exercise differs from ECG trace recorded at rest. Okay, so during exercises, you can see here, the rate of breathing increases, all right? Apart from the rate of breathing increasing, the pulse rate or the heartbeat rate also increases. Okay, when these things increase, we have increased rate of what? Respiration, so as to provide more energy for the activity or for the exercise. So the reasons to why there is a difference in recording of ECG trace at rest and at exercise is first that at exercise or during exercise there's increased breathing rate or pulse rate. During exercises there's increased pulse rate and heartbeats. Okay, so there's increased heart rates and what? And pulse rate. Okay, yeah. Okay, this allows more oxygen supply into the body. All right, then the oxygen is used for respiration. Is used for respiration to do what? To provide energy for muscle contraction. All right, for muscle contraction. Okay, also there is increased carbon dioxide concentration. All right, there's increased carbon dioxide concentration, which also causes increased breathing rate. All right, so these factors make the results or the traces recorded by the ECG during exercise to be different from the one that is recorded at rest. All right, so just one other way of monitoring activity of the heart. Okay, so we can count pulse rate, or we can listen to closing and opening of the valves. You place your fingers, let's say, on your hand or some part around your neck and listen to the closing and opening of what? Of the valves. All right, yes. So, or you can use the digital watch. All right, so here you can count pulse rate. Yeah, so these are ways to maintain or to monitor heart activity. B says, if exercise is very intense, an oxygen debt is formed. Complete the sentences. Oxygen debt results from buildup of dash in the dash during vigorous exercises. So oxygen debt usually results from building up of lactic acid. Okay, why lactic acid? Because glucose is not completely broken down, so it forms lactic acid. So it's built up of lactic acid in the blood. Okay, lactic acid is building up in the blood, all right, because there is incomplete breakdown of glucose, okay, during vigorous exercises. Outline how the body removes an oxygen debt. All right, so in order for the body to remove oxygen debt, so it means we need oxygen to completely break down the lactic acid by aerobic respiration. So first of all, we say that breathing rate remains high to provide high amount of oxygen. High amount of oxygen. All right, yeah, that's one thing. Then the lactic acid is carried in the blood to the liver. Okay, the lactic acid is carried to the liver, where it is broken down. It is broken down by oxygen, okay, which is definitely aerobic respiration. Okay, so it's broken down by oxygen, which is aerobic respiration. What? To form carbon dioxide and water. To form carbon dioxide and water, which means that it undergoes complete what? Respiration. Okay, yes, so that is how oxygen debt is removed from the body. Okay, a leaf can be defined or can be described as an organ. Describe an organ. So an organ, this is a group of what? Of tissues that perform a similar function. Okay, it's made up of tissues that perform what? A similar function. All right, so we say that it is a group of tissues that perform a specific similar function. Specific similar function. Yeah, that is an organ. B says figure 4.1 is a photomicrography of a cross section of part of a leaf. As we can see it, it's a part of what? Of the leaf. We have parts labeled A, B, and C. All right, A, B, and C. Okay, identify the tissues labeled A. All right, so these tissues over here, which kind of appear to have maybe like they appear like they have a long like structure, rectangular like structure. These tissues over here are usually known as the palisade cells. So we'll state here they are the palisade cells. Okay, identify the structures labeled C. So here C is this part over here. This part here usually represents the vascular bundle, which is made of xylem and phloem. So we'll say here this is vascular bundle. All right, describe how the tissues labeled B is adopted to maximize photosynthesis. The tissues labeled B here. Okay, so we have number 4B here, which says figure 4.1 is a photomicrography of a section part of the leaf. It has parts labeled, as we can see, A here, it has C over here, and it has B over here. All right, so identify the tissues labeled A. The tissue labeled A is here. This tissue is representing the upper epidermis. All right, so we'll state here this is upper epidermis. Okay, then identify the structures labeled C. The structures labeled C represent the vascular bundle. So we'll say here this is the vascular bundle, which is actually made of xylem and phloem tissues. And then describe how the structures labeled B is adopted to maximize photosynthesis. The structures labeled B are here. These are the palisade mesophyll cells. Now, how are they adopted to maximize photosynthesis? First of all, they have a lot of chloroplasts in them. All right, so the chloroplasts are arranged over here so that they can trap sunlight to allow the process of photosynthesis to take place. Okay, so we'll say here the tissues are palisade mesophyll cells. Okay, they are palisade mesophyll cells. All right, They are tightly packed and they contain chlorophyll that traps sunlight to be used in photosynthesis. Yeah, so this is what they have. They have lots of chlorophyll to allow them to conduct the process of photosynthesis. C says state two substances that are transported only in the phloem. Alright, so it means these are the products that have been made from the process of photosynthesis, right? Okay, what do we get from the process of photosynthesis? We get sucrose. One of them is sucrose. Okay, so sucrose is only going to be transported in the phloem. But also there are proteins, these smaller units of proteins, amino acids. They are also transported only in the phloem. So these food-like substances will only be transported in the phloem. Explain why some parts of a plant can act as both sink and source. Alright, so some parts will be used, alright, to provide or be used as a source of energy when a plant is growing, okay, and they'll be used to store, other parts will be used to store the products of photosynthesis. But at the same time, these parts, a source is a part of a plant that photosynthesizes, all right, and a sink is a part of the plant that stores the products of photosynthesis. Okay, so if a part is acting as a source, it means there are times that it is allowing or producing energy, all right. Let's say leaves. Leaves are like converting light energy into chemical energy, okay, which is photosynthesis process. And then that is when a plant is an adult. But when a plant is growing, it means the leaves are now growing from the plant, this young plant, a seedling. This time the leaf is not photosynthesizing, instead it's a sink. Now what is acting as the source? A seed, okay. Yes, a seed is now acting as a source, which means it's providing the necessary nutrients required by the plant. Okay, while at other times a seed or a root is acting as what? As a sink. When a plant is an adult, it means storing the products of photosynthesis. But there are other times it acts as a source, which means it's giving the necessary nutrients for what? For a plant. Okay, so we'll say they act as sink when the plant is growing, all right. The act as sink when the plant is growing, so this definitely is going to be the leaves, okay. The plant is growing, all right. Yeah, example the leaves, okay. And act as source during photosynthesis. All right, so example leaves, they act as sink when a plant is growing, all right, and then they act as source when a plant is photosynthesizing. So this is for an adult plant. You can use the growing seed, after the seed has broken up, the seed coats have broken out, so this seed is again acting as what? As a source, allowing the plant to grow. And when a plant has grown, it will be photosynthesizing, and you find that the products of photosynthesis are again going to be put into what? Into the seed. So at that time the seed will be acting as the sink, okay. This says the effect of carbon dioxide concentration on rate of oxygen production in aquatic plants was measured. A lamp was used to keep the light intensity constant. The oxygen gas released by the plant was collected in a gas syringe. The plant was placed in water that was kept constant at 20 degrees. Figure 4.1 shows the results. The rate of oxygen production was assumed to be the same as the rate of photosynthesis. Suggest why the rate of oxygen production was not the same as the rate of what? Of photosynthesis. Now, they assumed that it would be the same, but at the end of the day, the rate was not the same. Why? Because not all oxygen that is produced during photosynthesis will be released by the plant. Why? Because this oxygen is going to be used for the process of respiration by the plant. Okay, so at the end of the day, we'll say that it was not the same. It was not the same as some amount of oxygen is used by the plant in other processes like respiration. In other processes like respiration. All right. Yeah. So this could be the reason to why we didn't have the same amount of oxygen as rate of photosynthesis. All right. Yes. Then we have this graph over here which shows rate of oxygen production per meter square per second, and then we have concentration of carbon dioxide. All right, so you can see as carbon dioxide increased, rate of oxygen also increased until we reached a maximum rate. Okay, explain the results. Okay, so we go back to the graph. Now we have already seen that as carbon dioxide is increasing, the rate of oxygen is also doing what? Increasing. So it means that in order for oxygen to be produced, you must have carbon dioxide. Oh, in order for photosynthesis to take place, carbon dioxide must do what? Must increase, must be there. And of course, when we reach at a concentration of 500, the rate of oxygen produced remained the same. So here, carbon dioxide was at its what? Its maximum. So it was not affecting the rate of photosynthesis anymore. That's why the oxygen production remained 0.25. So we can say here that the limiting factor for photosynthesis is carbon dioxide. Okay, photosynthesis is carbon dioxide. Is carbon dioxide. All right, as carbon dioxide concentration increases, rate of oxygen production increases. Oxygen production increases. All right, increases. Okay, this shows rate of photosynthesis increases with carbon dioxide concentration. With carbon dioxide concentration. All right, yeah. Then after that we'll say at maximum volume of carbon dioxide, it is not limiting photosynthesis. It's not limiting photosynthesis, and the volume of oxygen produced remains constant. Remains constant. Yeah, so that is the description of that graph. Okay, the investigation was repeated with the same type of aquatic plant at 10 degrees centigrade. Draw a line to predict the results at 10. So at 10, it means that the rate of photosynthesis is now affected by temperature also. So what will be the rate of photosynthesis? Definitely, the rate of photosynthesis will decrease. So it can start from this point over here. All right, go and then be like that, because now temperature is also affecting the rate of photosynthesis. Okay, number 5 says measles is a transmissible disease. The percentage of population that were vaccinated against measles in a country was determined. The number of confirmed cases of measles in the country was also recorded. Figure 5.1 shows the data that were recorded between 1975 and 2010. So we can see here from the graph, percentage of population that was vaccinated kept on increasing with what? With time. As percentage of vaccinated people increased, also the percentage of number of cases did what? Decreased. All right, you can see here in 1975 we have percent of vaccinated people around 50%, and we can see that the rate of the number of people who had the disease was a bit high. Okay, yeah, so as the percentage of vaccinated people increased, the rate of the disease also decreased. Okay, so calculate the percentage change in number of cases of measles between 1980 and 1990. We come to a graph here and look for 1980. All right, here. Okay, number of cases. So what was the number of cases in 1980 here? All right, so which is approximately equal to 140,000. So we'll say 1980 we had 140,000 individuals. In 1990, what is the number of cases in 1990? 1990 is here. Alright, so you can see the number of cases in 1990 is a bit low. We can assume this one to be 14,000, alright, according to the scale. So 1990, the cases were 14,000. Right, so to get the percentage change, we'll take the reading of 1990, which was 14,000. Subtract the reading of 1980, which was 140,000. We'll divide to the reading of 1980, which was 140,000. And then the answer that we'll get over here, we'll multiply it to 100. So when you work out this one here, 14,000 minus 140,000, you get negative 126,000. Divide it to 140,000, you get 0.9. Now 0.9 multiply it to 100. This will give you 90%. But it's going to be negative percent to show us that the number of cases has decreased. All right. Yeah. So that is the percentage change within this particular time. Roman 2 says, describe the data that has been shown over there. Okay. So we have described the data that has been shown in that particular graph. All right. Yeah. Okay. So let's look at the graph here. Okay. We can see that the dotted line is representing percentage of population vaccinated, which has been increasing from 1975. It has been going up. And of course, the straight line is kind of representing the population or the number of cases that have measles. Now, we can see that as the number of vaccinated population increased, the number of measles kept on going down. So we are going to describe this data as follows. That the percent of vaccinated population increases from 1975, right? From 1975 it increases. Okay. As vaccinated population increases, the number of measles cases decreases. So as the number of many people being vaccinated is increasing, and then the number of people who are being vaccinated also does what? Decreases. All right. Yeah. Then here we can see that the number of cases for measles remain the same from 2000 to 2010, but the number of vaccinated population falls from 1995 to 2005. A lot of people were not vaccinated, and then again it rises in 2010. So we'll say the number of people vaccinated decreased from 1995 to 2005. Okay. So here the number of vaccinated population decreases from 1995. Then the number of measles cases remains the same from 1995 to 2010. Okay, from 1995 to 2010. Yeah, so this is a short description that we can see from the graph. And then explain how vaccination protects people against transmissible diseases such as measles. All right, so vaccination will involve a harmless pathogen. Okay. A harmless pathogen is going to be mixed with what? Within the medicine and then injected as a vaccine. Now, after it is injected, it will stimulate production of antibodies within the body, okay, which are going to fight this particular antibody. Now, after they have been produced, these antibodies will remain in the body as memory cells to protect the body. So in case a new infection occurs, it means this individual will already have antibodies that will protect them from what? From the infection. Okay? Okay. So say here a vaccine contains harmless pathogens of the disease. Okay? Then they result to production of antibodies antibodies specific to the pathogen, all right. Yeah, the antibodies destroy the pathogen and leave mammal cells. Destroy the pathogen and mammal cells are left behind. And mammal cells are kept providing long immunity against the pathogen. Immunity against the pathogen causing disease. Right, so mammal cells are produced because vaccines act as what? Active immunity. All right, they stimulate the body to produce antibodies specific to the particular kind of infection. So we can say that this is a form of active immunity as the antibodies are specific to the what? To the pathogens. These are specific to the kind of pathogens. Okay, yeah. So that is how a vaccine protects transmissible diseases. The human body has several defenses against pathogens. Stage 2 of the body chemical barriers to the pathogens. Okay, so what are chemicals that are produced by the body to fight against pathogens or to prevent pathogens from entering into the body? One is hydrochloric acid. Hydrochloric acid is there to what? To prevent pathogens or act as a chemical barrier. But also we have mucus. So these two here are acting as what? As chemical barriers. Okay, describe the process of blood clotting. So the process of blood clotting usually involves production of what? Of fibrinogen from blood platelets. So blood platelets produce for fibrinogen. Fibrinogen is a kind of protein which is then converted into fibrin. Then fibrin is going around the maybe cut area so that it can form what? A mesh to trap blood cells. And then this mesh-like feature which has now trapped red blood cells is usually known as what? A scab. So we'll say here blood platelets produce fibrinogen. Okay, a protein fibrinogen. Then fibrinogen is converted to fibrin. All right. Then fibrin traps the blood, the flowing blood. Okay, fibrin traps the blood. Okay, yes, to form a mesh. All right, so they form a mesh which actually traps what? Blood. Traps the blood forming a mesh, and this can actually be a scab. Number 6. 6A says, figure 6.1 shows a food web. State the number of trophic levels we can see on this particular food web. All right, so when you look at this food web, we have this trophic level 1, 2, 3. We have this one here 1, 2, 3, 4. We have here 1, 2, 3. Okay, so we have we can go 1, 2, 3, 4. So we have a maximum of 4 trophic levels. Right here 4. Then state the name of one organism that feeds at both 3rd and 4th trophic level. It feeds at both 3rd and 4th trophic levels. So come here. Corn, for example, mice 3. Hawk is at trophic level number 3. But then again, hawk is at 1, 2, 3, 4. It is at trophic level number 4. So hawk is feeding at both trophic levels. Light here, hawk. And then state the name of the type of energy that is transferred between trophic levels. All right. So the energy that is usually transferred between trophic levels is chemical energy. So we'll write here chemical. Right, chemical energy. B says the total biomass of snakes is much less than the total biomass of mice in the food web shown in figure 6.1. Explain why the total biomass of snakes is less than the total biomass of mice. All right, so we come back. If you look here, snakes are at a different trophic level from what? From mice. Okay, so if they're at a different trophic level from mice, it means they don't get the amount of energy that mice gets. Okay, so at the end of the day, we'll have less biomass in snakes in comparison to what? To mice. So we'll say here, snakes occupy a high trophic level than mice. Okay, trophic level than mice. This is the first thing. Okay, then energy is usually lost in between trophic levels. So energy is lost in between trophic levels. So the energy that is in mice will not reach what? The mice. Okay, so hence not enough energy reaches the snakes. Not enough energy reaches the snakes. All right, yeah. So at the end of the day, less energy will reach the snakes, and of course they will appear to have a less biomass in comparison to biomass of what? Of mice. C says food shortages that result in famine can be caused by many factors. Describe how drought can contribute to famine. So drought is a dry condition. It means no water and no rain, just no source of water. So if there's no water, we expect drying of crops, okay, if they are there. And if crops are not there, we expect to plant them. But when we plant the seeds will not be able to do what? To grow. Okay, but apart from that, if we have crops, we expect them to photosynthesize and they need water to do so. But if there's no water, it means they'll not photosynthesize. They'll end up drying and dying. Okay, wilting and dying. And of course, if they die, it means we will not have plants as a source of food. And we know plants are producers, all right? They can be used by being consumed directly, but apart from that, they're being consumed by other animals. Let's say a gazelle, for example. It feeds on plant, and then we have other animals that feed on a gazelle. So if plants dry out, it means there'll be no source of food for herbivores, there'll be no source of food for carnivores, and at the end of the day, that is going to be a famine. All right, yeah. So here we say drought is lack of water. Okay, it can cause drying of crops, and failure of photosynthesis, which will eventually also cause drying of what? Of crops. Okay, then at the end of the day, again, drought causes plants not to germinate, because we need water in order for the plant to do what? To grow. Hence we will not have what? Plants. Okay, not to germinate, and hence less amount of food for animals or maybe livestock, yeah, which in turn becomes famine. Okay, yeah. So this is what is happening during this process. Okay, yeah. So we can, yeah. All right. So yeah, that was the last question for this work. All right.